Hello, I'd like to extend a warm welcome from a rather cold Lübeck from the Department of Dermatology here at the University of Lübeck. I'd like to take a few moments to discuss and introduce our forthcoming publication in the British Journal of Dermatology. Let's go inside. My name is Dr. Langan and I'm a dermatologist in the Department of Dermatology in Lübeck, Germany, and an honorary lecturer at the University of Manchester in the UK. As the corresponding author of the review article entitled "The Role of the Microbiome in Psoriasis, Moving from Disease Description to Treatment Selection," which will be published in the forthcoming weeks in the British Journal of Dermatology, I would like to take just a few moments to outline the background to and key messages arising from the publication. Human skin is a surface area of almost 2 square meters and contains around 5 million hair follicles. Traditionally viewed as a protective physical barrier to pathogens and potential allergens, it's now clear that human skin is much more than just a wall separating us from the environment. In fact, the use of 16S ribosomal RNA sequencing has shed new light on the extent and diversity of the microbes with which we share our skin. Put simply, 16S ribosomal RNA sequencing technology now allows us to identify a range of bacteria based on the hypervariable regions of the prokaryotic 16S ribosomal gene. Moving away from traditional culture-based bacterial identification, we're now able to identify bacteria which we were previously unable to culture, and they went completely underappreciated in the cutaneous context. Combining simple skin swabs with highly sophisticated molecular sequencing techniques, complemented with state-of-the-art bioinformatics tools, the bewildering complexity of the cutaneous microbiome is now becoming apparent. Indeed, perturbations of this complex ecosystem, termed dysbiosis, may play a role in inflammatory skin disease, as has been postulated in the case of chronic common inflammatory skin diseases such as psoriasis, a disease associated with considerable morbidity and which affects 1 to 2% of the European population. Several studies have now identified shifts or changes in the composition of the resident skin microbiota, leading some authors to propose that specific cutaneotypes exist based on which bacterial phyla, genera and species are most prominent. The extent to which psoriasis-specific microbiome signatures exist and how they can be utilised to predict response to treatment is under investigation. Our review synthesises the largely descriptive published literature examining the cutaneous microbiome in psoriasis and calls for an integration of cutaneous microbiome signatures with comprehensive geno and phenotyping of patients and their diseases. Perhaps the holy grail of microbiome research is the skin site-specific modulation of resident bacterial populations in order to treat and or even prevent skin disease. Whether this can be achieved directly via the application of topical agents or indirectly via immune modulation or even harnessing crosstalk with other microbiomes, for example the gastrointestinal microbiome as part of the gut-skin axis, remains an area of intense speculation. In the age of personalised medicine, the integration of clinical data with individual microbiome profiles may facilitate treatment selection and consequently optimise treatment efficacy, bringing next-generation sequencing into today's dermatology clinics.